Hello everyone. This video is for BSc program 6th semester paper 8 biochemistry. This is session number 28 from biological oxidation chapter, and the topics covered in this session are substrate level phosphorylation and very important mitochondrial electron transport chain and oxidative phosphorylation. So you are going to learn what is substrate level phosphorylation, in detail about electron transport chain and oxidative phosphorylation. Now, phosphorylation is an addition of a phosphate group. To be more simpler way of defining, in this phosphorylation what happens is you are going to synthesize ATP from ADP in the presence of an enzyme ATP synthetase or ATP synthase. That is, the synthesis of ATP is known as phosphorylation. That is, when ADP combines with inorganic phosphate in the presence of an enzyme, ATP is synthesized. That is, you are introducing a phosphate group to ADP known as phosphorylation. Now, there are different types of phosphorylation reactions. Which are they? Substrate level phosphorylation and oxidative phosphorylation. So you will be asked the definition of these two with the examples. That is, what is substrate level phosphorylation? Explain with an example. And what is oxidative phosphorylation? The substrate level phosphorylation, which is also known as SLP, it is the phosphorylation where ATP is synthesized by the addition of a phosphate donated by a high energy molecule is known as substrate level phosphorylation. See here, these are the high energy phosphate molecules. High energy phosphate molecules. They donate their phosphate group. Here to synthesize ATP. The product is ATP. So the high energy molecules which possess high energy and donate a phosphate for the synthesis of ATP in substrate level phosphorylation is called as high energy phosphate. The entire process is known as substrate level phosphorylation. Coming to specific examples. So you have to explain substrate level phosphorylation with examples. So this is phosphoenol pyruvate. PEP is phosphoenol pyruvate. In presence of an enzyme pyruvate kinase, it transfers its phosphate group to ADP. In turn, ATP is synthesized. And phosphoenol pyruvate is converted to pyruvate, which you have learnt in glycolysis. Okay. So, phosphoenol pyruvate donates its phosphate group to ADP. In the presence of an enzyme pyruvate kinase, a molecule of ATP is synthesized. When in turn this PEP is converted to pyruvate, this reaction is known as substrate level phosphorylation. This reaction is example for substrate level phosphorylation. This is example 1. Coming to example 2. See 1,3-bisphosphoglycerate or 1,3-diphosphoglycerate. See there are two phosphate groups. One of the phosphate group is transferred to ADP. In the presence of phosphoglycerate kinase, ATP is synthesized and this itself is converted into 3-phosphoglycerate, 3-PG. This is also one of the important step in glycolysis. So these two examples are for substrate level phosphorylation. One is conversion of phosphoenol pyruvate to pyruvate. Second one is conversion of 1,3-biphosphoglycerate into 3-phosphoglycerate. So when a question is asked, explain substrate level phosphorylation with an example, you have to define what is substrate level phosphorylation and you should be able to write the examples simply, not in words. You should know to write the reaction. So please practice writing the equations by writing many times. Otherwise, you may not be able to reproduce the reactions properly. Okay. And one more example is creatinine phosphate converts to creatinine. Here also phosphate group is transferred to ADP. In turn ATP is synthesized is an example for substrate level phosphorylation. Now coming to second one, oxidative phosphorylation or electron transport chain. So it's synthesis of ATP during transport of electrons from NADH2 to oxygen is known as oxidative phosphorylation. ಏನಂತ ಕರೀತೀರಾ? Oxidative phosphorylation. Okay. Now, oxidative phosphorylation, where does it occur? It occurs in the electron transport chain. Oxidative phosphorylation occurs in the electron transport chain. Okay. Now, what is this ETC? So ETC is a series of electron carriers arranged in the increasing order of reduction potential. So I'll tell you what are these electron carriers. Remember, in electron transport chain, all these electron carriers are arranged in the increasing order of reduction potential. Okay. In the last session, I have explained you regarding this. So reduction potential is the ability of a molecule to accept electron. It's an electron affinity which is again measured in terms of reduction potential, and it is measured in terms of electron volts. So electron reduction potential is the ability of a molecule to accept electrons. So as we move along the chain, the ability of electron carriers to accept electron increases. The ability of this electron carriers to take electron increases or to accept electron increases. So, it is this reduction potential that enables movement of electron through the chain. So, before we go in detail about electron transport chain, remember there are a series of electron carriers here. There are a series of electron carriers here. And how are these electron carriers arranged? They are arranged in the increasing order of reduction potential. Okay. And as we move along the chain, the ability of electron carriers to accept electron increases. Okay. Where is this located now? See, the electron carriers of ETC are all located embedded in the inner mitochondrial membrane of every cell. So now you know synthesis of ATP during transport of electrons from NADH2 to oxygen in presence of electron carriers is known as oxidative phosphorylation. See, in the previous which I explained substrate level phosphorylation, there were no electron carriers at all. That was only a simply transfer of a phosphate group to ADP to synthesize ATP. But whereas when you come to oxidative phosphorylation, I am bringing a word here, electron carriers. So the synthesis of ATP during electron transport, that is the transport of electrons from NADH2 to oxygen, where the ATP is synthesized with the help of electron carriers is known as oxidative phosphorylation. Okay. And where are these electron carriers located? They are located in the inner mitochondrial membrane of every cell. See here, this is the outer mitochondrial membrane, this is cytosol, and this is the inner mitochondrial membrane, and this is the mitochondrial matrix. So this is the site of electron transport chain. Now, let me explain you components of ETC. So before explaining the components of ATP, see, there are energy-rich molecules like glucose or fatty acids, which are metabolized by a series of reactions which you have already learned. And ultimately, they yield carbon dioxide and water. See, most of these reactions are dehydrogenations in which one or two hydrogen atoms are transferred from substrate to a hydrogen acceptor. Substrate to a hydrogen acceptor. These hydrogen acceptors are specialized coenzymes. These hydrogen acceptors are what? Specialized coenzymes associated with the dehydrogenase enzymes. And of these, NAD+, FAD, FMN are the most common electron carriers. They are the most electron common electron carriers. And the reduced coenzymes in turn donate pair of electrons to a specialized set of electron carriers, and this is collectively known as electron transport chain. See, as electrons are passed down the electron transport chain, what happens is they lose much of their energy, and a part of the energy is trapped in the form of ATP. It is trapped in the form of ATP by phosphorylation of ADP, which is known as oxidative phosphorylation. So as I told, the electron transport chain is present in the inner mitochondrial membrane and is the final aerobic pathway by which electrons derived from various oxidizable metabolites flow to oxygen, and electron transport and ATP synthesis proceeds continuously in all cells of the body that contain mitochondria. Now you know what is the importance of this electron transport chain. Okay. Now, what are the components of ETC? So the electron transport chain has a series of electron carriers. Sometimes they'll ask you for 2 marks this only. Write the electron carriers of electron transport chain. So electron carriers of ETC are NAD, FMN, FAD, coenzyme Q, and a family of cytochromes. I repeat, electron carriers of ETC are NAD, FMN, FAD, coenzyme Q, and a family of cytochrome. Now, you should understand the reduced state of NAD is NADH2. So, many times we write it as NADH plus H plus. Okay. And the oxidized state is NAD plus. Reduced state of FMN is FMNH2 or FMNH plus H plus. And oxidized state is FMN plus. Come to FAD. So, what will be the reduced state? FADH2. What will be the oxidized state? FAD plus. Coming to coenzyme Q, reduced state is COQH2 and the oxidized state is COQ. Coming to family of cytochrome, so these are the class of proteins which contain heme as the prosthetic group. So, you know, heme in the cytochromes contain iron. It contains iron, and the reduced state for this is Fe2 plus, oxidized state is Fe3 plus. And the cytochromes present in the ETC are cytochrome B, C1, C, A plus A3. Okay. So, which are the components of ETC or the electron carriers of ETC? NAD, FMN. I have also explained you how will be the reduced state and what will be the oxidized state for all this. So, the electron carriers of electron transport chain are NAD, FMN, FAD, coenzyme Q, and a family of cytochromes which contain heme as the prosthetic group. This contains iron, reduced state is Fe2 plus, and the oxidized state is Fe3 plus. And the cytochrome present in ETC are cytochrome B, C1, C, A and A3. Only if you understand this reduced state and the oxidized state, you will be able to write the diagrammatic representation of electron transport chain. Now, see, the electron carriers of ETC are not present as a continuous chain. Instead, they occur as 4 discrete protein complexes in the inner mitochondrial membrane. Protein complex 1 contains NAD and FMN electron carriers. Protein complex 2 contains FAD electron carrier, and protein complex 3 contains cytochrome B, cytochrome C1 electron carriers, and protein complex 4 contains cytochrome A and then A3 electron carriers. Okay. Now, see, this is how they are arranged. The sequence of electron carriers in ETC are arranged this way. See, what is this substrate? See, you have learnt already now what is glycolysis, Krebs cycle, urea cycle, etc. The substrate that donates electrons to NAD include whatever the intermediates you get or the final products you get: pyruvate, alpha ketoglutarate, isocitrate, malate, glutamate, and beta hydroxy acyl CoA are the reduced substances. And those that donate electrons to succinate dehydrogenase include succinate and then fatty acyl CoA. Okay, so this is how the sequence of electron carriers runs in ETC, NAD, FMN or NADH, dehydrogenase, COQ, coenzyme Q, in complex 3, cytochrome B, cytochrome C1. Here you have cytochrome A and then A3. And finally, we come to molecular oxygen. Okay. Now, this is how the sequence of electron carriers are arranged in electron transport chain. So, to write the electron transport chain, you should know this order. Only if you know the order, you can clearly write the diagrammatic representation of electron transport chain. Now, I have to explain you in detail how the reaction takes place. Now, we'll start from here. You know NADH2 is the first electron carrier. It loses 2H plus and 2 electrons. So when NADH2 loses 2H plus and 2 electrons, what happens? It undergoes oxidation to form NAD plus. It undergoes oxidation to form NAD plus. Now, the 2 electrons thus liberated are added to FMN, which is the next electron carrier with a higher reduction potential. And what happens to FMN? It's reduced to FMNH2. Okay. Since oxidation of NADH2 is oxidized to what? NAD plus. FMN is, and the reduction of FMN to what? FMNH2 occurs simultaneously. A redox couple is generated at the junction of NAD and FMN. See, in the previous slide or the previous session, I told you what is a redox couple, what are conjugate acid-base pairs. You should know and you should recall that everything, and also what is oxidation and reduction in terms of electrons. Then only you understand electron transport chain. Now I'll come to the next step. See, this FMNH2 loses 2H plus and then 2 electron and undergoes oxidation to what? FMN. The 2 electrons thus liberated are added to what? FAD. Remember the sequence. The next electron carrier with the higher reduction potential, and FAD gets reduced to FADH2. Now, since oxidation and reduction FMNH2 and FAD occur simultaneously, a redox couple is generated at the junction of FMN and then FAD. Okay. Come to the next step. This FADH2 loses 2H plus and then 2 electron and undergoes oxidation to what? FAD plus. See, in the very previous slide I told you what is the reduced form and then oxidized form. Please remember. The 2 electrons thus liberated are added to what? To the next electron carrier. What is the next electron carrier? Coenzyme Q. And what happens to coenzyme Q? Coenzyme Q gets reduced to COQH2. Since oxidation and reduction occur simultaneously here, again a redox couple is generated at the junction of FAD and then COQ. So remember, what is the first electron carrier? NADH2, FMNH2, FADH2. Okay. Now, I'll come to the next step. See, this coenzyme QH2 loses 2H plus and then 2 electrons, undergoes oxidation to what? COQ. Thus the 2 electron liberated are added to what? Come to the next one, family of cytochromes, Fe3 plus of cytochrome B, which is the next electron carrier with the higher reduction potential, and Fe3 plus gets reduced to Fe2 plus. So this generates a redox couple at the junction between coenzyme Q and then cytochrome B. Similarly, redox couple between every 2 consecutive electron carriers, namely cytochrome B, cytochrome C1, cytochrome C, C, and then cytochrome A, A3 are generated. Okay. At each junction, iron is oxidized from Fe2 plus to Fe3 plus and gets reduced from Fe3 plus to Fe2 plus. What happens ultimately? 2 electrons liberated from cytochrome A3 is added to oxygen and 2H plus finally to synthesize one water molecule. Now, since the ETC involves transfer of electrons from NADH2 ultimately to what? Oxygen for the synthesis of water molecule, this process is known as oxidation. Okay. The 2H plus liberated at every redox couple is pumped into perimitochondrial space, that is a space between outer and inner mitochondrial membrane. So this leads to a charge separation that generates a force called proton motive force. This proton motive force intensifies along electron transport chain. See, it may look difficult for you when I explained you this oxidation reduction, but first, you understand to learn ETC, what are the electron carriers, what is the reduced form and what is the oxidized form. Then, a series of oxidation reduction reactions occurs, and finally, two electrons liberated from cytochrome A3 are added to half O2 and then 2H plus. Ultimately, oxygen, this gets reduced to water. Okay. Since ETC involves a transfer of electron from NADH2 to oxygen for the synthesis of water, this process is known as oxidation. Okay. Now, see, this proton motive force thus generated is used to synthesize ATP from ADP, okay, and PI. ATP is generated from ADP and PI at three junctions. I will show you the three junctions also. See, at the redox couple between NAD and FMN in protein complex 1, one ATP is generated, first ATP. At the redox couple between cytochrome B and then cytochrome C1 in protein complex 3, second ATP is generated, and the third ATP is generated at the redox couple between cytochrome A and then cytochrome A3 in protein complex 4. Okay. Since ATP is synthesized from ADP and PA, this is known as phosphorylation. The very first slide I told you what is phosphorylation. Okay. As phosphorylation occurs simultaneously with oxidation along the ETC, it is known as oxidative phosphorylation. Okay. The phosphate of the ATP synthesis in oxidative phosphorylation is obtained from cytoplasm, and the enzyme responsible for the synthesis of ATP from ADP and PI is ATP synthetase. Okay. So, I'll show you how ATP are generated at each level, at different level in ETC. See, to calculate the energetics, we have taken 1 NADH2 when enters into electron transport chain, 3 ATP molecules are synthesized, and 1 FADH2 when enters into electron transport chain, 2 ATP synthesized, because this FADH2 bypasses one step. I'll show you in electron transport chain diagram. That's why for 1 FADH2, 2 ATP molecules are synthesized at the end of oxidative phosphorylation, and for 1 NADH2, 3 ATP molecules are synthesized at the end of oxidative phosphorylation. See, this is the schematic representation of electron transport chain. I told you the different electron carriers. NAD, FMN, coenzyme Q, family of cytochrome. Ultimately, electrons are accepted by this oxygen and which gets reduced to water. And which are the ATP site generation? See here the first ATP synthesized from ADP and then inorganic phosphate. So here the second ATP is generated and here the third ATP is generated. So when NADH2 or NADH and then H plus, the number of ATP generated when enters into electron transport chain is, you see here, 1, 2 and then 3. When FADH2 enters into electron transport chain, which bypasses one step, the number of ATP generated 1 and then 2, because this you will take to calculate the energetics of glycolysis, Krebs cycle and then beta oxidation pathway. So here you see, so from the reduced metabolites, the reaction starts from NADH2 to NAD. So here you can see this NADH2 getting oxidized to NAD plus. In turn, these electrons are accepted by Fmn and getting reduced to FmNH2. So this series continues here. This series continues. So here there is a continuous oxidation and reduction of this Fe2 plus and then Fe3 plus. Fe2 plus getting oxidized to Fe3 plus, getting reduced to Fe2 plus, oxidized to Fe3 plus, getting reduced to Fe2 plus in the family of cytochromes, and then ultimately these electrons are accepted by oxygen and which gets reduced to water. Since the ATP is generated here through a process of electron carriers. Remember, they are all arranged in the increasing order of the reduction potential. Okay, so electron flows in this direction. Ultimately, oxygen is the electron acceptor which get reduced to water. Since ATP is generated through electron through the use of electrons or through electron carriers, this is also known as oxidative phosphorylation. And for one molecule of NADH, 3 ATPs are generated. For one molecule of FADH, there is generation of only 2 ATP molecules when they enter into electron transport chain. Okay, so here you should know where does this electron transport chain is located, where it occurs, which are the electron carriers, and how in sequence these electron carriers are arranged, and how the redox couples are generated, how the redox couples are formed, and which are the sites of ATP generation in electron transport chain. Okay, so this session you have learned what is substrate level phosphorylation, what is oxidative phosphorylation, what are the electron carriers in electron transport chain, and in detail how the reactions or how the redox reactions occurs ultimately to generate ATP in electron transport chain. You have to write and practice this electron transport chain and the electron carriers, then only you can reproduce, because sometimes they'll ask you for 6 marks. They'll ask you for 6 marks this, write the schematic representation of ETC. So to write the schematic representation of ETC, you should know these electron carriers. So here, since it is a definite question for you for the examination, so please remember this slide, sequence of electron carriers in ETC. So once you know how the electrons are carried away in ETC in this order, then you can easily write the redox couples, the redox reactions, and ultimately you can show the site of ATP synthesis. Thank you.